Imagine a cancer cell, right?
Mhm.
It's perfectly camouflaged, just sitting right there in your tissue.
Right.
But it's completely invisible to your immune system.
Yeah.
Now, I want you to imagine your white blood cells acting like, well, microscopic safecrackers.
Oh, I like that analogy.
Yeah, they're dropping off these self-destructing flash drives that literally force that cancer cell to unmask itself. I mean, it sounds like complete science fiction, right?
It really does. It sounds like something straight out of a spy novel, honestly.
Yeah.
And, for a long time, we had absolutely no idea this mechanism even existed.
Which is exactly why we are jumping into this today. Welcome to the Deep Dive, everyone. We are unpacking a truly groundbreaking 2026 article from the journal Cancer Cells.
Yeah, by Mengying Hu and her colleagues.
Exactly. And when I tell you this is a total paradigm shift in how we understand the immune system, I am seriously not exaggerating. This paper completely unravels a brand new way that our T cells communicate. We're going to go through exactly how they act at delivery trucks and, how they use this mechanism to turn the tables on invisible tumors.
Yeah, to really appreciate how wild this discovery is, you have to look at what the biology textbooks have said for the last, few decades.
Right.
We've basically been operating under the assumption that T cells, which are kind of the assassins of the immune system, that they only communicate in two main ways.
It was always thought to be either direct physical contact, like touching another cell.
Yes.
Or by secreting those soluble proteins into the fluid around them.
Exactly. Soluble proteins like cytokines. Specifically, we usually talk about interferon gamma.
Like a chemical scent trail.
Yeah, exactly. But this new research proves that activated T cells are doing something so much more complex. They are actively packaging and basically throwing tiny lipid bubbles, which we call extracellular vesicles, and they're stuffing them with actual genomic DNA to reprogram the surrounding cells.
Okay, let's unpack this dense paper, starting with those bubbles, because the delivery system itself is just fascinating.
It really is.
We're talking about extracellular vesicles, or EVs, just so we're all on the same page. What exactly are these things?
Well, pretty much every living cell in your body releases EVs.
Okay.
You can think of them as tiny, 150 nanometer lipid balloons. They just sort of pinch off from the cell's outer membrane and float around in the extracellular space.
Little biological soap bubbles?
Yeah, perfect. But the researchers in this study, they didn't just look at any random EVs. They specifically focused on T cells, and what they noticed was a massive difference between a resting inactive T cell and an activated one.
And an activated T cell is one that has, recognized a threat, right? It's geared up for a fight.
Precisely. So in the lab, they took T cells and stimulated them with activation beads and growth factors like IL-2.
Okay.
And they collected the vesicles from these cells during their peak expansion phase. They called them activated T cell-derived EVs, or ATEVs.
ATEVs, got it.
Right. And then they compared those to normal vesicles from non-activated T cells, which they called anti-EVs.
And the difference in how these ATEVs behave in a living body is just staggering.
Oh, completely night and day.
Right, because when the researchers isolated these active vesicles, they tagged them with a near-infrared fluorescent dye and then injected them into healthy mice. And they didn't just float around randomly like you'd expect.
No, they didn't. Normally, if you inject standard EVs into a bloodstream, say from a skin cell, they mostly just get swept up by the body's filtration system.
Like the liver?
Yeah, they pool in the liver or the spleen. But these ATEVs had an incredible, highly specific homing algorithm.
A homing algorithm.
Yeah. In healthy mice, they completely swarmed the immune organs, particularly the lymph nodes. And when they ran the exact same experiment in mice that had tumors, the ATEVs actively bypassed normal, healthy tissue and homed straight in on the tumor microenvironment.
They were actively seeking it out. But wait, how do they actually know where to go? A lipid bubble doesn't have a brain. It doesn't have a GPS.
Right.
It has to be a chemical interaction, right?
Exactly. It all comes down to the proteins that are decorating the outside of that bubble. The researchers ran a comprehensive proteomics analysis.
They're taking an inventory.
Right, taking an inventory of literally every protein on the surface of these ATEVs. And they found these vesicles were heavily coated in very specific adhesion molecules.
And the heavy hitters here were a protein called fibrinogen and an integrin complex called LFA1.
Yes, LFA1, which is made of ITGAL and ITGB2. And those specific proteins are designed to lock onto a very specific receptor called ICM1.
ICTAM1.
Now, here is the crucial piece of context for you. ICM1 is an inducible cell adhesion molecule. It isn't just expressed everywhere all the time.
Okay.
Healthy, quiet tissues, like your brain or your heart, they have almost zero ICAM1 on their surface. But in areas of inflammation like a lymph node gearing up for an immune response, or a highly inflamed tumor ICAM1 expression just goes through the roof.
So it's like the tumor's accidentally painting a giant bullseye on itself.
Exactly.
So if we want to visualize this, think about the ATEV as a smart bomb drone. And the LFA1 and fibrinogen on its surface, that's like the hook side of a piece of Velcro.
I love that.
And the ICAM1 on the lymph nodes or the cancer cells is the fuzzy landing pad. So the drone can float right past a smooth, healthy heart cell, but the second it brushes up against a fuzzy, inflamed cancer cell, it sticks instantly.
That is a perfect way to visualize it. And the researchers actually proved this was the exact mechanism using a really elegant experiment.
Yeah.
They injected the mice with a blocking antibody that specifically covered up the ICAM1 receptors on the tumors.
Oh, basically they just put tape over the fuzzy side of the Velcro.
Exactly. They taped over it.
And what happened?
The homing completely failed.
Wow.
Yeah, the ATEVs just circulated blindly, and eventually they just got cleared by the liver. They never made it to the tumors. So the ICM1 interaction is absolutely essential.
I mean, I love when an experiment gives you such a clean proof of causality, but I do have to ask the logical question here.
Sure.
Wait, why would a T cell send a vesicle instead of just migrating there itself? I mean, isn't that like mailing a letter to someone you live with? Just go talk to them.
It's a great question, but you have to consider the massive scale of a tumor microenvironment.
Okay.
What's fascinating here is that a single T cell physically can only interact with one, maybe two cells at the exact same time. It has to physically touch them.
Right.
We call that contact-dependent signaling. It's a huge bottleneck. But by releasing thousands or even tens of thousands of these ATEVs, a single T cell can suddenly amplify its voice. It allows for contact-independent communication, or what we call juxtacrine signaling from a distance. One activated T cell in a lymph node could potentially alter the behavior of thousands of cells deep inside a tumor it has never even physically touched.
So it's transitioning from a one-to-one conversation to a 1 to 10,000 broadcast.
Precisely. It's an exponential amplification of the immune signal.
That makes so much sense. Okay, let's do a quick recap for you to lock this first part in.
Let's do it.
Number 1, T cells send out ATEVs, like targeted biological drones. Number 2, they use a homing algorithm based on Velcro-like proteins, specifically LFA1 binding to ICAM01. Number 3, this allows a single T cell to broadcast a signal to thousands of targets at once.
Perfect summary.
And here is a question for you to think about.
Yeah.
Think about a disease you know of where immune cells struggle to physically reach the target. How could an ICAM1 targeted delivery system like this completely change the game for treatments?
That is a really exciting thing to ponder.
Right. Okay, so we have these targeted drones, and they know exactly where they're going. But, you know, a delivery truck is really only as good as its cargo.
Very true.
What are they actually dropping off when they get to the tumor?
This is where the paper really shifts the paradigm. The researchers used highly sensitive sequencing techniques, RNA sequencing and whole genome sequencing, to crack open these vesicles and look at the payload.
And the assumption, based on previous EV research, was probably that they'd find some proteins, maybe some transient messenger RNA.
Exactly. Bits and pieces of short-lived genetic material. But what they actually found was a massive enrichment of double-stranded genomic DNA.
Wait, actual dsDNA?
Actual dense structural double-stranded DNA. And it wasn't just a random chopped-up assortment of the T cell's genome either. It was highly specifically enriched with antigen processing and presentation genes.
We call those APP genes.
Right. Genes like MHC class 1 and 2, but also the machinery genes like TAP1 and PSMB8.
Let's break down why that specific cargo is so incredibly important for the listener. Antigen presentation is essentially how a cell shows the immune system what's going on inside it, right?
Oh, exactly.
It uses those MHC molecules to put little protein fragments, antigens, on its surface, sort of like displaying items in a shop window.
That's a great way to put it. If a cell is healthy, it displays normal proteins, and the immune cells just patrol right past it. If a cell is infected by a virus, or if it's mutated into a cancer cell, it displays those abnormal proteins in the window, and the immune system attacks.
But cancer cells are incredibly sneaky. One of the main ways they become invisible, like we talked about at the very beginning, is by hiding those display windows.
Yes, exactly.
They mutate and downregulate their MHC molecules.
Yes.
If there's no window, the immune system can't see the mutated proteins hiding inside. So these T cells are packaging the exact genetic blueprints the APP genes needed to force a cell to rebuild those display windows.
Yes. They are literally forcing the tumor to make itself visible again. But, in science, just finding DNA inside a vesicle doesn't automatically mean that DNA is doing the heavy lifting.
Right, you have to prove it, because there are proteins and RNA floating around in there too. So how did they isolate the DNA and prove it was the active ingredient?
They used a chemical called baseline zero DNase, or DNA zero. You can think of it as a highly specific molecular shredder.
Okay.
It only digests DNA. It leaves RNA and proteins completely untouched. So they took the ATEVs, ran them through this shredder, and then applied them to dendritic cells and tumor cells.
Let me guess. The magic completely stopped.
It completely vanished. The recipient cells did not upregulate their MHC molecules.
Yeah.
The immune response was totally flat.
Wow.
Yeah, without intact double-stranded DNA, the drones were completely useless. The DNA is definitively the active ingredient.
That is incredible. But when I was reading through the sources, I noticed there was a major, major hurdle they had to clear with reviewers.
Oh, the cellular debris argument.
Yes. A lot of critics in this field argue that when you find DNA outside of a cell, it's just garbage. It's just debris from dead or dying cells that happen to get wrapped up in a lipid bubble by accident. How do you prove this isn't just a biological trash bag?
That was a massive point of contention they had to address. If a T cell dies and ruptures, its DNA splatters everywhere. To prove this was an active, intentional manufacturing process, the team used a chemical tag called BRDU.
And BRDU is basically a synthetic building block of DNA, right?
Exactly. It's a nucleotide analog. A living cell will only incorporate BRDU into its DNA if it's actively building new strands of DNA at that exact moment in time.
Okay, so it tags new construction.
Right. So the researchers added BRDU to the environment of the activated T cells for a short period. They call it a pulse chase experiment. They pulsed it and then washed it away.
So any DNA built during that specific window lights up with the tag?
Precisely. And when they analyzed the DNA inside the released EVs, it was heavily tagged with BRDU. Meanwhile, the old existing genomic DNA resting inside the T cell's nucleus had very little of the tag. Oh, wow.
Yeah. This absolutely proved that the evDNA wasn't old trash from dead cells. The T cells were actively firing up a molecular printing press, synthesizing brand new genomic DNA specifically to package into these drones.
Okay, but wait. Pushback time. If I rip a page out of my own instruction manual and hand it to you, well, I don't have that page anymore.
Right.
If a T cell is actively printing and giving away its genomic DNA, especially genes critical for its own immune function, doesn't it deplete its own blueprint? Does the T cell damage itself to send this signal?
That is the exact logical leap the researchers made too, and they tested for what we call copy number variation.
Okay.
They found that the T cells undergo a localized amplification process. They don't cut the genes out of their genome. They make extra copies of those specific EPP genes.
Oh.
Yeah, their own genomic integrity remains perfectly intact. They're just printing extras for distribution. Okay, time for another quick recap to lock this in. Number 1, these drones are loaded with newly minted double-stranded DNA. Number 2, this DNA specifically contains the instructions for antigen presentation, or APP. Number 3, destroying the DNA makes the drones useless. And number 4, the T cells print extra copies so they don't harm themselves.
Spot on.
And here's a question for you listening at home. If your cells are constantly shedding EVs and DNA into your bloodstream, what kind of secret genetic messages do you think your own cells are passing around right now? It's kind of wild to think about.
It really makes you look at blood in a whole new way.
Totally. So we know T cells recognize a threat, they ramp up production of specific genes, package this newly minted DNA into Velcro-coated vesicles, and unleash thousands of them. That is the delivery. But getting the package to the door is only half the battle.
Right, because once that drone fuses with the tumor cell and dumps its DNA into the cytoplasm, we have a huge biological problem.
A massive problem. In cellular biology, free-floating DNA in the cytoplasm is the ultimate red flag.
Yes, it is.
Because healthy DNA belongs locked safely inside the nucleus. If it's floating in the outer fluid of the cell assumes it's being invaded by a virus.
Precisely. And mammalian cells have a very sensitive alarm system for this, known as the CGAS sting pathway. Normally, if this pathway detects cytosolic DNA, it slams on the emergency brakes.
Right.
It triggers a massive inflammatory shutdown, halts protein production, and often forces the cell to undergo apoptosis, which is programmed cell death.
So how on earth is this T-cell DNA bypassing the alarm? If the goal is to get the tumor cell to read the DNA and build new display windows, tripping an alarm that shuts down protein production defeats the entire purpose.
It does. And the researchers actually assumed at first that the ATEVs were working by triggering this melod alarm, maybe causing a general inflammatory response.
Sure, that makes sense.
But they tested it by using sting knockout cells. These are cells that literally do not have the alarm system genetically.
And the ATEVs still worked.
Perfectly. The antigen presentation still went up. They also used chemical inhibitors like A151 and JK inhibitors to block other DNA sensors and interferon pathways. Nothing stopped the ATEVs. The immune boost was definitely not coming from standard inflammatory alarms or IFN gamma signaling.
Which means the DNA isn't just sitting in the cytoplasm setting off alarms. It's somehow sneaking into the vault. It has to be getting inside the recipient cell's nucleus to actually be read and turned into proteins.
Exactly.
But the nucleus is heavily fortified. It's surrounded by a tough nuclear envelope made of structural proteins called lannins. How is the T-cell DNA breaking in?
This might be the most fascinating mechanism in the entire paper. When they went back to that proteomics data, the inventory of proteins inside the vesicle, they found it was packed with a protein called granzyme B.
Granzyme B. But wait, usually granzyme B is a weapon.
Yes.
Cytotoxic T cells use it to execute infected cells. They punch a hole in the cell and inject granzyme B to force the cell to commit suicide.
Exactly. It's traditionally known as an executioner protein, but context and dosage are everything in biology.
Okay.
Here, it isn't being used to kill the cell. Delivered in small, precise amounts inside these vesicles, granzyme B acts as a molecular scalpel. It specifically cleaves those laminin proteins you just mentioned.
No way.
Yeah, it literally cuts a temporary hole in the nuclear envelope of the recipient cell.
Here is where it gets really interesting. It's a literal heist.
It is.
The ATEV is a safecracker. Granzyme B is the blowtorch that cuts a precise hole in the nucleus. And the DNA just slips right inside.
And it doesn't just go anywhere inside. The researchers tracked it and found it localizes specifically to the euchromatic regions of the host's chromatin.
Euchromatic.
Yeah, these are the loosely packed areas where active gene transcription is currently happening. It slips right into the cell's active reading room.
Okay, but this raises a truly terrifying possibility.
You're going to say.
If you have foreign genomic DNA entering the nucleus of a cell, does it splice itself in? Are we permanently mutating the cancer cell? Because if we are permanently altering genomes, that has massive, massive safety implications for any future therapy.
The researchers were hyper aware of this risk. To find out, they used something called long-read nanopore sequencing.
Okay. Explain how that differs from normal sequencing.
Standard sequencing chops DNA into tiny fragments to read it quickly, which can make it hard to tell if two pieces are genuinely fused together. Nanopore sequencing reads extremely long, continuous strands of DNA as they pass through a tiny pore.
So you can see the whole sentence, not just the individual words scattered around.
Right. They looked at thousands of these long reads to see if the evDNA had integrated into the host's genome, and the integration rate was 0.
0.
Completely indistinguishable from the background noise of the control group. There is no permanent genomic integration.
So it's what biologists call episomal transcription.
Yeah.
If we go back to our safecracker analogy, the DNA is like a Mission Impossible cassette tape. It gets dropped into the safe, the cell plays the message which says, show your antigens, the cell builds them, and then the tape dissolves. The original genome of the cell remains totally untouched.
And if we connect this to the bigger picture from an evolutionary perspective, it is an absolute masterstroke.
Why is that?
Well, why wouldn't the immune system want this to be a permanent change? Because if a T cell permanently mutated an antigen-presenting cell, that cell would never stop presenting those antigens. It would constantly trigger the immune system long, long after the original threat was gone.
Which would lead to a massive, unending autoimmune disease.
Exactly. Transient expression allows the body to fight off an immediate, temporary threat without leaving a permanent genetic scar that could cause autoimmunity down the road.
Nature basically figured out non-permanent gene therapy millions of years ago. It's humbling. Okay, let's recap the heist. Number 1, the mechanism evades the usual CGAS sting alarms. Number 2, granzyme B acts as a blowtorch to let the DNA into the nucleus. Number 3, the DNA is read temporarily, episomally, leaving no permanent genetic scars.
Perfect.
So here's a question for you. Imagine using this exact granzyme B mechanism in human medicine. How could a non-permanent gene therapy delivery system change the way we treat genetic diseases without the risk of permanent mutations?
That's the billion-dollar question right there.
It really is. All right, so we know the molecular mechanism, but what does this actually look like in a living, breathing biological system? Like, can this actually melt a tumor?
This is where we move from the Petri dish to real clinical translation. The team used syngeneic mouse models.
Meaning mice with fully intact normal immune systems.
Exactly, so they could observe the real-world interactions. And they didn't make it easy on themselves. They chose some of the most notoriously deadly cold tumors in oncology.
Right, cold tumors. These are the cancers we mentioned earlier that have completely scrubbed their MHC molecules. The immune system looks right past them. They used models like SB28, which is a vicious glioblastoma, or brain cancer.
Yeah, and KBCY for pancreatic cancer and PIMT for breast cancer. Historically, these tumors do not respond to standard immunotherapies because the T cells literally cannot see them to attack them.
Right.
But when the researchers intravenously injected the ATEVs into these mice, the ATEVs used that ICAM1 homing algorithm, bypassed the healthy tissue, and swarmed the cold tumors.
They dropped off the granzyme B blowtorch, delivered the DNA, and forced the invisible cancer cells to sprout those MHC display windows.
Suddenly, the tumors were waving a massive red flag. The immune system woke up. Endogenous T cells, the mouse's own T cells and natural killer cells rushed into the tumor microenvironment.
But tumors don't just give up. They are highly adaptable. I know that in the brain cancer model, the ATEVs alone significantly slowed the tumor growth. But in the pancreatic and breast cancer models, the tumors started fighting back, didn't they?
They did. Once the tumors realized they were being attacked by T cells, they deployed a defensive shield called PD-L1.
Okay.
This is a protein that basically binds to a receptor on T cells called PD-1, and it exhausts them. It basically tells the attacking T cells to go to sleep. So the ATEV monotherapy eventually hit a wall.
But that's a problem we already know how to solve in modern medicine. We have drugs called checkpoint inhibitors, specifically anti-PD-1 antibodies, that block that exact exhaustion signal.
Exactly. So the researchers combined the natural ATEVs with the clinical anti-PD-1 drug, and the synergy was explosive.
Wow.
Anti-PD-1 alone does almost nothing to these cold tumors because, well, there are no T cells there to begin with.
Right. You can't keep a T cell awake if it's not even there.
Exactly. But the ATEVs turned the cold tumors hot, pulling the T cells in. Then the anti-PD-1 drug kept those T cells awake and fighting. They saw dramatically prolonged survival in these mice.
And the histological analysis, like looking at the actual tissue of what happened inside the tumor, is mind-blowing. They didn't just find a scattered bunch of immune cells. They found the creation of tertiary lymphoid structures, or TLS.
This is a huge concept in cutting-edge oncology. A tertiary lymphoid structure is essentially a miniature, temporary lymph node that the immune system builds directly inside an inflamed tissue.
It's like the immune system realizing the front line is too far from headquarters, so it builds a forward operating base right in the trenches of the tumor.
That's a perfect analogy. The ATEVs recruited B cells, follicular dendritic cells, and T cells, and organized them into these structured fighting bases deep behind enemy lines.
That's wild.
And the presence of TLS is actually one of the strongest predictors of long-term survival in human cancer patients.
Which brings us to the crucial point. This isn't just an artifact of mouse biology, it translates to humans.
Yes. To validate this, they acquired freshly dissected primary tumor explants from human glioma and pancreatic cancer surgeries.
So actual human tissue.
Actual human tissue, and they found a direct positive correlation. The human tumors that naturally contain more of this evDNA also had significantly higher levels of natural T-cell infiltration. The core biology holds up completely across species.
So what does this all mean? Bringing this all together, what is the future here? Are we looking at a completely new wing of the oncology clinic? I know right now, one of the most advanced treatments we have is CAR T-cell therapy,
Right.
where we extract a patient's T cells,
Yeah.
genetically engineer them, and put them back. It is miraculous, but it's incredibly expensive. It takes weeks, and it can cause severe, sometimes fatal side effects like cytokine release storms.
CAR T is powerful, but you're right. Dealing with living cells is a logistical and medical nightmare. The ultimate hope here is what we call acellular immunotherapy.
Therapy without the living cell, just using the vesicles.
Exactly. Imagine if we could mass produce these targeted ATEVs in a bioreactor. You wouldn't need to harvest the patient's own cells at all. You could have an off-the-shelf biological drug that homes directly to inflamed tumors, bypasses the inflammatory alarms, and delivers a temporary genetic code to make the tumor visible. It would perfectly prime the tumor for existing checkpoint inhibitors.
It functions as an incredible, natural, non-viral delivery vehicle.
Okay.
Of course, we have to acknowledge the reality of fungal translation. We are not there tomorrow.
No, definitely not. Manufacturing extracellular vesicles at scale is still notoriously difficult. We have to figure out exact dosing parameters, purification, how frequently it needs to be administered. There's a lot of work to do.
We have a long road of clinical trials ahead.
We do. But this mechanism, this stealth delivery of genomic DNA via granzyme B, is just a biological goldmine.
Okay, time for our final recap. Number one, in living models, this combination with immunotherapy literally melts away notoriously deadly invisible tumors. Number two, it does this by building immune bases, those TLS, inside the tumors. And number three, it turns immunologically cold tumors hot, which is highly synergistic with existing drugs.
Nicely summarized.
So final question for you. If we can bottle these ATEVs as an acellular drug, do you think it will eventually replace traditional CAR T-cell therapies entirely? Something to think about.
Definitely.
This paper completely reframes how we view the immune system. We started this deep dive talking about biological communication as a clean, simple lock and key system. But looking at how a T-cell actively prints DNA, packages it into a drone, lobs it across a tumor, blowtorches a nucleus, and temporarily rewrites the cell's identity. The reality is so much more chaotic and so much more brilliant than we ever gave it credit for.
It really is. And, I want to leave you with one final unscripted thought to really let all of this marinate.
Okay, let's hear it.
We've spent this entire time marveling at how we can harness this mechanism to boost the immune system and cure cancer. But in biology, every powerful gas pedal usually has a shadow side.
Oh, okay. Where are you going with this?
Consider the exact opposite scenario. What if autoimmune disease, conditions where the body relentlessly attacks its own healthy tissues, what if they are actually caused by a glitch in this very system?
Why, really?
Yeah. What if an overproduction of this evDNA is the root cause? If hyperactive T cells are constantly throwing these genetic flash drives around, forcing normal, perfectly healthy cells to needlessly sprout antigen display windows.
Oh, wow.
Could that be the spark that ignites diseases like lupus or rheumatoid arthritis? If we can understand how granzyme B opens the safe to turn immunity on, maybe we can learn how to lock it down.
Using the exact same pathway to turn off immunity instead of turning it on, that flips the entire script. It makes you wonder how many other secrets are hiding in the muddy waters of our biology, just waiting for us to sequence them.
Absolutely.
Well, thank you so much for joining us on this deep dive today. Keep questioning the consensus, let these concepts marinate, and we will catch you on the next deep dive.